Okay, hello everyone. In this small video, we will be talking about galactose metabolism. So, this whole topic you might expect in your 2 markers case based question or reasoning question, or I guess that can be asked in your 5 marker short questions also. Okay, so let us start with the topic. Galactose metabolism. Almost exclusively the galactose is metabolized by liver. Okay, this is a very important point. Okay, so ultimate byproduct of the galactose metabolism is a glucose. Okay, so galactose का glucose में conversion होता है liver में. Almost exclusively it is metabolized in the liver. So why the galactose is important? The importance of the galactose is it can synthesize so many byproducts. That is very important. That is, first is lactose, second one glycosaminoglycans, third is cerebrosides, fourth one glycolipids, and some glycoproteins also. This you have learned in your carbohydrate chemistry chapter. Okay, let us understand the galactose metabolism. Okay, first key enzyme that you should know is a galactokinase, which converts galactose to galactose 1-phosphate. Here in this step, the ATP is utilized. 1 ATP is utilized per 1 molecule of galactose. Okay, now these galactose, with the help of this rate limiting enzyme, that is your galactose 1-phosphate uridyl transferase, which can be converted to glucose 1-phosphate, and in this reaction 1 molecule of UDP glucose is going to be converted into UDP galactose. G for glucose and UDP GL means it is a galactose. Okay, this ultimately UDP galactose is acted upon by epimerase enzyme, and this ultimately UDP galactose is again converted into UDP glucose. This UDP glucose is actually stored as a glycogen, and upon requirement this glycogen is going to be break down, and that will be a formation of glucose. So this is called glycogenolysis. This process is called glycogenolysis. Okay, what happens in a classical case of galactosemia, that I will be discussing later in this part also, but having just a highlight, in the classical case of galactosemia, we have complete deficiency of galactose 1-phosphate uridyl transferase enzyme, which is a rate limiting enzyme. Okay, this is you should know. Sometimes these UDP galactose, which is acted upon by galactose 1 pyrophosphate enzyme, ultimately going to be converted into galactose 1-phosphate. Okay,